This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to today's exploration of a fascinating and somewhat unsettling phenomenon, human brain shrinkage. Over the next few minutes, we'll delve into why our brains naturally decrease in size as we age, what factors can accelerate this process, and most importantly, what science reveals about keeping our minds resilient. We'll also examine some surprising discoveries about brain health that challenge conventional wisdom. Whether you're curious about cognitive longevity or simply fascinated by the mysteries of the human mind, this narration offers insights that might just change how you think about thinking itself. Chapter 1. The Reversal. 6 million years ago, our ancestors split from the lineage that would become chimpanzees. At that time, our forebears possessed brains roughly the size of a modern chimp's brain, measuring around 350 cubic centimeters in volume. This ancestral brain would fit comfortably in a pint glass. Today, the average human brain measures approximately 1,350 cubic centimeters, nearly a pitcher's worth of neural tissue. This dramatic expansion, a near quadrupling in size, represents one of the most striking transformations in human evolution. Yet this story of relentless growth contains an unexpected twist that challenges our assumptions about progress and evolution. The expansion of the human brain followed a complex trajectory across multiple hominin species in Africa, Europe, and Asia. Australopithecus afarensis, the species that includes the famous Lucy fossil, lived between 3 and 4 million years ago, with a brain of roughly 450 cubic centimeters. This represented a modest increase over earlier hominins like Sahelanthropus and Ardipithecus, but these early relatives still possessed brains well within the range of modern great apes. They walked upright, used their hands with increasing dexterity, and likely possessed rudimentary social behaviors, but their neural architecture remained fundamentally ape-like. The real acceleration began with the emergence of the genus Homo roughly 2 million years ago. Homo habilis and Homo rudolfensis, among the earliest members of our genus, pushed average brain volumes to around 680 cubic centimeters. These species left behind the first unambiguous stone tools, simple flakes and choppers that required planning, fine motor control, and the ability to envision a finished product from raw material. Their larger brains supported these cognitive demands in ways that Australopithecus brains apparently could not. Homo erectus, a species that persisted for over 1 million years and spread from Africa across much of the Old World, achieved average brain sizes of 950 cubic centimeters. This steady enlargement continued with Homo heidelbergensis, whose brains approached 1,200 cubic centimeters roughly 400,000 years ago. These hominins represented intermediate stages in the trajectory toward modern brain size, each increment bringing neural processing power closer to what we possess today. They controlled fire, constructed complex tools, hunted large game cooperatively, and almost certainly communicated through some form of language. Their expanding brains made these accomplishments possible. The Neanderthals, our closest extinct relatives who lived in Europe and Western Asia until about 40,000 years ago, possessed remarkably large brains. Individual Neanderthal skulls reveal cranial capacities ranging from 1,172 to 1,740 cubic centimeters, with many specimens exceeding the modern human average. These robust hominins, adapted to the harsh glacial climates of Ice Age Europe, developed sophisticated technologies, buried their dead with apparent ceremony, cared for injured and disabled group members, and created symbolic art. Their barrel chests, massive jaws, and enormous teeth demanded substantial neural processing simply to manage the biomechanics of their muscular frames. The brain regions controlling motor function occupied more space in Neanderthal skulls than in ours. Yet for all their neural capacity, Neanderthals went extinct. The last populations vanished from their final refuges in southern Iberia and the Balkans roughly 40,000 years ago, leaving behind only trace amounts of DNA mixed into the genomes of modern Eurasian populations. Their larger brains did not save them when climatic shifts pressured their populations, when competition with expanding Homo sapiens groups intensified, or when their social networks proved too small to maintain viable populations across fragmenting habitats. This extinction creates a profound puzzle. If brain size determined evolutionary success, Neanderthals should have outcompeted their smaller-brained Homo sapiens contemporaries. They did not.
Then came the Cro-Magnons, early modern Homo sapiens, who appeared in Europe around 40,000 years ago. These were our direct ancestors, anatomically modern humans who nevertheless differed from us in striking ways. Fossil evidence reveals that Cro-Magnons possessed the largest brains in human evolutionary history. Their skulls housed brains averaging close to 1,500 cubic centimeters, with some individuals reaching 1,600 cubic centimeters or more. These Ice Age humans stood at the peak of a 2-million-year trend of increasing brain size across the genus Homo. The Cro-Magnons who painted the caves of Lascaux and Altamira possessed brains roughly 10% larger than modern humans. They crafted sophisticated tools from stone, bone, and antler. They created elaborate personal ornaments, carved figurines, and painted vivid images of animals on cave walls deep underground. They hunted woolly mammoths across the frozen steppes of Europe, tracked reindeer herds through seasonal migrations, and harvested salmon from rivers swollen with glacial melt. Their physical remains show they were taller than modern humans, more robust, with heavier bones and more pronounced muscle attachments. Everything about them was larger, including the skull that housed their brain. But something changed. The trajectory that had pointed upward for millions of years reversed itself. Modern human brains measure, on average, about 1,350 cubic centimeters. We have lost brain volume equivalent to a tennis ball compared to our Cro-Magnon ancestors. For men specifically, average brain volume has declined from roughly 1,500 to 1,350 cubic centimeters. Women's brains have shrunk by similar proportions. When exactly this reversal occurred remains debated, as explored in the next chapter.
This reversal represents something unprecedented in the story of human evolution. Homo naledi and Homo floresiensis, small-brained species that survived into surprisingly recent times, remind us that brain size did not always follow a simple upward trajectory across all hominin lineages. Homo naledi, discovered in South African caves and dated to between 335,000 and 236,000 years ago, retained a brain of only 500 cubic centimeters, despite being contemporary with early large-brained Homo sapiens. Homo floresiensis, the diminutive hobbit of Flores Island in Indonesia, persisted until perhaps 50,000 years ago with brains of just 400 cubic centimeters, smaller than a chimpanzee's. These small-brained species challenge simplistic narratives about inevitable brain expansion leading to human dominance. They survived for hundreds of thousands of years with neural equipment a fraction of ours. Yet their pattern differs fundamentally from ours. They maintained small brains inherited from small-brained ancestors. They represent stable alternative strategies, not reversals of prior expansion. We are the only hominin species to experience such pronounced brain growth followed by significant contraction. Alone among our relatives, living and extinct, we climbed to a peak and then descended from it.
Comparing modern humans with our extinct relatives reveals the full scope of this transformation. When we stand today beside a reconstructed Neanderthal skeleton in a museum, we tend to focus on the differences in facial features, the robustness of the bones, the barrel chest. But hidden inside that distinctive skull sat a brain that, in many cases, exceeded our own in sheer volume. The same holds true for our direct ancestors, the Cro-Magnons. These Ice Age humans who buried their dead with elaborate ceremony, who crafted sophisticated tools and jewelry, who navigated vast territories without maps, possessed larger brains than any human alive today. This creates a puzzle that echoes across multiple dimensions of our understanding. If bigger brains enabled the evolutionary success of the genus Homo, if increasing cranial capacity defined the trajectory from Australopithecus to Homo erectus to Homo heidelbergensis to Homo sapiens, why did the trend reverse? What changed in recent human history to make smaller brains advantageous, or at least not disadvantageous enough to prevent their reduction?
The question becomes even more intriguing when we consider cognitive performance. Larger brains do not necessarily equate to greater intelligence. Neanderthals, with their impressive cranial capacities, went extinct while smaller-brained Homo sapiens thrived and spread across the globe. Today, individual variation in brain size among healthy adults shows little correlation with measures of cognitive ability. A person with a 1,200 cubic centimeter brain can achieve the same intellectual feats as someone with a 1,400 cubic centimeter brain. The size of the container matters less than what happens inside it, how efficiently the neural networks operate, how effectively different regions communicate, how the architecture organizes information processing. Yet the scale of reduction we are discussing extends beyond normal individual variation. We are not talking about small fluctuations of a few percentage points, but a systematic decrease of 10 to 15% across all human populations over a relatively brief span of evolutionary time. This represents a significant evolutionary change, one that must have either provided some advantage or at least not imposed enough disadvantage to be selected against.
When researchers examine the preserved skulls of our ancestors and extinct relatives, measuring the internal capacity of crania, they can calculate brain volumes with reasonable accuracy. These measurements, compiled from hundreds of specimens across multiple species and time periods, reveal the grand arc of human brain evolution. From the chimp-sized brains of our earliest hominin ancestors, through the steady expansion across the various Homo species, to the peak achieved by late Pleistocene Homo sapiens, and then the recent decline, each skull tells part of this story. The reduction has not been uniform across all brain structures. Some regions appear to have decreased more than others. The frontal lobes, associated with planning and executive function, show particularly pronounced changes. The temporal lobes, involved in language processing and memory, have decreased in volume. The hippocampus, crucial for forming new memories, has measurably shrunk. The cerebral cortex, the wrinkled outer layer where much of our thinking occurs, has thinned. Yet despite these anatomical changes, modern humans have achieved cognitive feats that would have seemed impossible to our larger-brained ancestors. We split atoms, mapped genomes, and sent spacecraft beyond our solar system. The pattern of brain evolution across the genus Homo reveals our uniqueness with stark clarity. In Homo habilis, Homo erectus, Homo heidelbergensis, and the Neanderthals, brains expanded. These species followed the trajectory that characterized human evolution for 2 million years. Bigger bodies supporting bigger heads, housing bigger brains with greater processing capacity. They used this capacity to survive in diverse environments, develop increasingly sophisticated technologies, and spread across much of the Old World. In Homo sapiens, after following this trend for most of our existence as a species, brains contracted. We alone reversed the pattern. This makes us unique among our relatives, the only hominin to turn back from the path that had defined human evolution since our genus first emerged. The implications ripple through our understanding of what drove human evolution and what makes our species different from all others that preceded us. The comparison with Neanderthals proves particularly illuminating, precisely because it confounds simple explanations. They possessed larger brains than we do. They developed sophisticated technology, including specialized tools that required complex multi-step manufacture. They created symbolic art, suggesting abstract thought.
They cared for group members who could not contribute to subsistence, suggesting empathy and moral reasoning. By many measures, they were as cognitively sophisticated as contemporary Homo sapiens populations in Africa and the Near East. Yet Neanderthals lived in smaller groups with less extensive social networks. Archaeological evidence suggests their populations remained fragmented across the landscape, with limited long-distance exchange of goods or ideas. When researchers analyze the stone tools found at Neanderthal sites, they find mostly local materials. When they examine the shells and other decorative items, they find few examples of materials transported over long distances. Neanderthal social worlds appear to have been more local, more isolated, more dependent on what individuals and small groups could accomplish independently. Homo sapiens groups, in contrast, maintained connections across vast distances even during the late Pleistocene. Shells from the Mediterranean appear at sites hundreds of kilometers inland. Obsidian from specific volcanic sources shows up at locations requiring days or weeks of travel to reach. Stone tool styles spread rapidly across regions, suggesting communication and cultural transmission between groups. These extensive networks meant that knowledge and innovations could spread, that populations could maintain genetic diversity despite low densities, and that groups could draw on resources and information from well beyond their immediate territory.
Perhaps this difference in social scale mattered more than individual brain size. A Neanderthal needed to be more cognitively self-sufficient because their networks provided less support. Each individual had to maintain more knowledge, develop more skills, and solve more problems independently. This pressure might have maintained selection for larger brains, even as Homo sapiens populations, with their more extensive social networks and richer cultural transmission, could begin reducing brain size without losing cognitive capability. The fact that brain reduction occurred after Neanderthal extinction, as modern humans spread globally and population growth accelerated, supports this interpretation. We did not need our brains to be as large because we could rely more heavily on collective knowledge, cultural transmission, and specialized expertise distributed across social networks. The cognitive burden shifted from individual to collective, allowing biological evolution to reduce the metabolic expense of maintaining large brains. What changed in recent human history to reverse a trend that had persisted for millions? Why do we, alone among our hominin relatives, have brains that grew and then shrank? The fossils document what happened. They show us the before and after, the larger skulls of the Pleistocene giving way to the smaller craniums of the Holocene. They reveal that modern humans possess less neural tissue than our immediate ancestors, and far less than our Neanderthal cousins who vanished 40,000 years ago. But fossils cannot directly tell us why. They preserve bone, not behavior. They show size, not function. They document change without revealing cause. For explanations, we must turn to other evidence, to the timing of reduction and what else changed during those same millennia, to studies of how brains actually function and what determines their size, to comparisons with other species who never experienced this reversal, and to theories about what makes human cognition unique among all primates. These questions lead us forward into subsequent chapters, where the focus shifts from documenting what occurred to understanding why it happened and what it means. Chapter 2. When and how much? When exactly did human brains begin to shrink? This question has provoked considerable scientific debate, with researchers proposing timelines spanning from 100,000 years ago to as recently as 3,000 years ago, a range so vast it essentially spans from deep prehistory to the dawn of recorded history. The answer matters because timing determines which environmental, cultural, or biological factors could plausibly explain the phenomenon. If brain reduction began 100,000 years ago, we must look to forces operating when Neanderthals still thrived, and modern human populations remained scattered and small. If it started just 3,000 years ago, we are examining something that occurred during the Bronze Age, within the rise of complex civilizations. Ian Tattersall, a paleoanthropologist at the American Museum of Natural History, has argued that brain shrinkage commenced approximately 100,000 years ago. This timeline situates the change far back in human prehistory, when Homo sapiens were just beginning to expand beyond Africa. Tattersall suggests this period marked a fundamental shift in how humans processed information, a transition from what he terms brute force thinking to symbolic information processing. Yet this early date conflicts with the fossil record. Skulls from Homo sapiens who lived 100,000 years ago do not consistently show reduced brain volumes compared to earlier specimens. Many fossils from this period reveal cranial capacities well above modern averages, fully within the range expected from the upward trajectory that had characterized human evolution for the preceding 2 million years.
More recent analyses using advanced statistical techniques have challenged this early timeline. Jeff Morgan Stibble, a cognitive scientist from the Natural History Museum in California, conducted a detailed study of 298 Homo sapiens skulls spanning the past 50,000 years. His analysis identified brain shrinkage beginning approximately 17,000 years ago, coinciding with the end of the last ice age when global temperatures rose and ice sheets retreated. Stibble's work revealed a striking correlation between brain size and climate. The warmer the climate, the smaller the brain. The cooler the climate, the larger the brain. This relationship suggested that brain reduction might represent an adaptive response to warming temperatures. The human brain, despite comprising only about 2% of body mass, consumes over 20% of resting metabolic energy, making it the largest energy hog of all organs. In cooler climates, larger brains produce more metabolic heat as a byproduct of their intensive energy consumption, potentially providing thermoregulatory advantages. As global temperatures warmed after the last glacial maximum, this heat production became less beneficial or even disadvantageous, possibly favoring individuals whose smaller brains generated less excess heat while still meeting cognitive demands. Yet even this relatively recent 17,000-year timeline may not capture the full story. In 2021, Jeremy De Silva and his team at Dartmouth College applied sophisticated change point analysis to a massive data set of 985 fossil and modern human crania. The statistical technique they employed specifically identifies moments when rates of change shift significantly, not gradual drifts, but discrete breakpoints in evolutionary trajectory. What they found surprised even the researchers. The analysis detected 3 major change points in hominin brain evolution. 2 represented periods of brain expansion, one at 2.1 million years ago with the early evolution of the genus Homo, and another at 1.5 million years ago, aligning with the spread of Homo erectus. These expansions were expected. The 3rd change point told a different story. De Silva's analysis placed the beginning of brain size reduction at approximately 3,000 years ago, give or take 1,000 years. This extraordinarily recent date situates the change during the Bronze Age. 3,000 years ago, Egyptians were building temples at Karnak, Greeks were developing their alphabet, Olmec civilization flourished in Mesoamerica, and the Zhou dynasty ruled China. This was not remote prehistory, but a time of sophisticated civilizations, written languages, and monumental architecture. The finding proved immediately controversial. Mark Grabowski and Bernard Wood challenged De Silva's conclusions, arguing that the dataset suffered from severe temporal imbalance. Of the 836 specimens De Silva analyzed that post-dated the emergence of Homo sapiens, 578 came from the most recent time period, essentially modern humans from the past few thousand years. This concentration of recent specimens, Grabowski argued, could create an artificial appearance of recent change when the reduction might actually have begun much earlier, but was simply less well documented in the sparser earlier fossil record. When Grabowski reanalyzed the data accounting for this imbalance, he found evidence suggesting reduction might have begun as early as 32,000 years ago, rather than 3,000. Yet De Silva's finding gained unexpected support from independent research using entirely different data.
Mathieu Henneberg, working with over 14,000 skull measurements spanning recent millennia, concluded that most of the decrease occurred during the last 3,000 years, precisely matching De Silva's timeline despite using a completely different dataset and analytical approach. This convergence of independent evidence strengthened the case for a genuinely recent and rapid reduction rather than a methodological artifact.
The debate over timing reflects fundamental methodological challenges in analyzing ancient skulls. Some studies rely on measurements of individual brain regions, while others calculate total cranial capacity. Some analyze specific geographic populations, while others attempt global assessments. The scarcity of well-preserved fossils from key time periods creates gaps in the data, particularly from Africa, where much human evolution occurred, but preservation conditions often prove challenging. A skull from 15,000 years ago discovered in Europe cannot easily be compared to one from 25,000 years ago found in Asia, given natural variation in brain size across populations. Despite these challenges, certain patterns emerge consistently. The largest-brained Homo sapiens specimens date to between 20,000 and 30,000 years ago, representing the peak of a trend building for millions of years. From this high point, brain volumes began to decline. Whether the reduction began 100,000, 20,000, or 3,000 years ago remains contested, but the direction of change does not. By the present day, modern human brains measure smaller than those of late Pleistocene ancestors. The magnitude of this reduction varies by study and time period. Analysis of more than 12,000 skulls from Europe and North Africa spanning the last 10,000 years revealed that male cranial capacity decreased by 157 cubic centimeters, a 10% reduction. Female skulls showed an even more pronounced decline of 261 cubic centimeters, representing 17% of brain volume. These figures represent averages across populations, with substantial individual variation. Some recent individuals possessed brains well within the ancestral range, while others fell significantly below. In China, analysis of 718 male skulls from the Holocene documented a 7.2% reduction in cranial volume from the Bronze Age to present. The rate of reduction appeared to accelerate after 3,500 years before present, a finding that aligns remarkably with De Silva's 3,000-year change point. This acceleration during the Bronze Age suggests that whatever forces drove brain size reduction intensified precisely when complex agricultural societies were consolidating and expanding. Similar patterns emerged from East Asia and Australia, where researchers documented approximately 10% reduction in cranial capacity, and from sub-Saharan Africa and Japan, where comparable trends appeared. If De Silva's 3,000-year timeline proves correct, the implications transform our understanding of brain reduction from a slow evolutionary drift into a rapid recent change unfolding across just a few millennia. His team calculated that brain reduction occurred at a rate 50 times greater than the previous increases during the Pleistocene. This acceleration suggests something changed dramatically in the human condition around 3,000 years ago, though skull measurements alone cannot reveal what that change might have been.
The global pattern of reduction provides additional clues. The fact that brain size decreased across all populations on all continents suggests a common cause rather than localized selective pressures. The fact that it occurred recently, within the past few thousand to 20,000 years, depending on which timeline proves correct, points toward environmental or cultural factors that changed universally for human populations during this period. And the fact that reduction accelerated dramatically, especially if the 3,000-year timeline holds, indicates powerful selective pressure operating over a remarkably brief span. The magnitude alone tells us this change mattered biologically. Losing 10 to 17% of an organ's mass represents a significant evolutionary event. Evolution typically does not discard expensive tissue without reason, suggesting brain reduction must have either provided direct advantages or occurred because the selective pressure maintaining large brains had weakened. The scientific controversy over precise timing will likely persist as new fossils emerge and analytical techniques refine.
But the fossil record has established the foundation. Human brains grew for 2 million years, and then, during the most recent fraction of our existence as a species, began to contract. The change point occurred sometime between 100,000 and 3,000 years ago, with growing evidence favoring the more recent end of that range. The reduction proceeded at rates far exceeding earlier expansion, affecting all populations globally and reducing brain volumes by 10 to 17% depending on sex and population.
What forces could drive such sustained, directional change in a trait as fundamental as brain size? The timeline itself constrains the possibilities. Whatever caused this reduction must have changed universally for humans during the late Pleistocene and Holocene, must have operated powerfully enough to overcome the selective forces that had driven expansion for 2 million years, and must have provided advantages or, at minimum, imposed costs insufficient to prevent the change despite its magnitude. The skull measurements document what happened. Understanding why it happened requires looking beyond the fossils themselves to the environmental, social, and cultural changes that transformed human life during these same millennia.
Chapter 3: Survival of the Friendliest. When humans began selectively breeding wolves into dogs roughly 15,000 years ago, the animals changed in predictable ways. Their teeth became smaller, their snouts shortened, their ears flopped. They developed patches of white fur. Their temperaments shifted toward docility and friendliness, and their brains shrank. Domestic dogs possess skulls 10 to 15% smaller than wolves of equivalent body size, housing brains that weigh proportionally less. This reduction occurred not through deliberate breeding for smaller brains, but as an inadvertent consequence of selecting for tameness. Across domesticated species, from sheep to cats to foxes deliberately bred for docility in Siberian experiments, the pattern repeats. Selection for pro-social behavior produces animals with smaller brains than their wild ancestors. Could humans have done this to themselves? The self-domestication hypothesis proposes that Homo sapiens underwent a process analogous to animal domestication, but driven by social selection within our own species rather than imposed by external breeders. As human groups grew larger and social complexity increased, individuals who could cooperate, tolerate close proximity to non-kin, control aggressive impulses, and coordinate with others gained reproductive advantages. Those who were too aggressive, too intolerant, or too antisocial faced exclusion, punishment, or reduced mating opportunities. Over thousands of generations, this social selection pressure favored pro-social traits in ways that parallel how human breeders favored tameness in wolves. The physical markers align suggestively. Modern human faces are smaller and less robust than those of our Pleistocene ancestors. Our teeth have decreased in size. Our skulls show reduced sexual dimorphism, with males less dramatically larger than females compared to earlier hominins. Our skeletons overall display what biologists term juvenilization or neoteny, the retention of juvenile characteristics into adulthood. These traits match the suite of changes seen in domesticated animals, collectively termed domestication syndrome. The fact that human brains also reduced during this same period, shrinking by 10 to 15%, mirrors exactly the brain reduction observed across domesticated species. Anthropologist Brian Hare, studying dog cognition and evolution, developed what he termed the survival of the friendliest hypothesis to explain this pattern. The phrase inverts the traditional survival of the fittest to emphasize that, in social species operating in large groups, friendliness and cooperation can matter more than raw competitive ability. A wolf that excels at dominance, fights, but cannot tolerate close pack coordination will be outcompeted by wolves that sacrifice some individual prowess for social cohesion. Apply this logic to humans living in increasingly large groups, and you get a selection regime where the most cognitively dominant individuals might be outcompeted by more cooperative, more socially skilled, more tolerant individuals who could function effectively in dense settlements. The mechanism operates through neural crest cells during embryonic development.
These cells give rise not just to brain structures, but also to facial features, pigmentation patterns, hormone-producing organs, and behavioral circuits. Selection for reduced fear and aggression responses alters neural crest cell migration patterns, which cascades into changes across multiple systems. A gentler temperament comes packaged with a shorter snout, smaller teeth, and often a smaller brain. You cannot easily select for one without getting the others. This explains why domestication syndrome appears as a coordinated suite of traits rather than isolated changes. Hormonal systems link these changes mechanically. The same genetic and developmental pathways that regulate stress hormones like cortisol and adrenaline also influence brain growth, facial development, and behavior. Selection for individuals who remain calm in social situations, who can tolerate crowding without aggressive responses, who cooperate rather than compete, necessarily alters these hormonal systems. The resulting hormonal profiles affect not just behavior, but also the physical structures of the brain and face during development. Smaller brains become a byproduct of selection for prosocial temperament, rather than a direct target of selection.
Archaeological evidence shows accelerating settlement size and population density during the transition to agriculture and beyond. Hunter-gatherer bands typically number 25 to 50 individuals, small enough that everyone knows everyone personally and most interactions involve kin. But as humans settled in permanent communities, group sizes expanded dramatically. This required unprecedented tolerance for proximity to non-kin, ability to cooperate with strangers, and capacity to navigate complex social hierarchies without constant violent conflict. Individuals who could thrive in these dense social environments left more descendants than those who could not. A person with aggressive temperament might excel in small hunter-gatherer bands where dominance hierarchies were relatively simple. But in a crowded agricultural village where coordinating with dozens of households determined success, that same aggressive temperament became a liability. Social exclusion, punishment by coalitions, or simply reduced access to cooperative networks would reduce reproductive success. Over generations, this pressure shifted the population toward individuals with more docile, more prosocial, more cooperative temperaments. The cultural evolution work of anthropologist Joseph Henrich provides crucial context for how this self-domestication became possible. Henrich argues that human success depends not on individual cognitive brilliance, but on accumulated cultural knowledge transmitted across generations. No individual invented agriculture, metalworking, or writing from first principles. These technologies emerged through countless small innovations compounding over centuries. Each generation inherited the accumulated discoveries of predecessors, made modest improvements, and passed refined versions to descendants. This cumulative cultural evolution meant that group-level knowledge far exceeded what any individual could develop alone. Consider the kayak used by Arctic hunters. This sophisticated watercraft represents centuries of refinement. The design optimizes hydrodynamics for hunting in frigid waters. Construction requires specialized knowledge of wood selection, sealskin preparation, bone tool manufacture, and waterproof stitching. No single person invented it. Each generation inherited designs, experimented with modifications, and transmitted improvements onward. The accumulated micro-innovations across many generations produced technology no individual could conceive independently. This pattern of cumulative cultural evolution transformed what natural selection valued in individual humans. In a world where survival depends on individual cognitive ability to solve novel problems, selection favors large brains with substantial processing capacity. But in a world where survival depends on learning from others, cooperating effectively, and contributing to collective efforts, selection can favor social skills over raw cognitive horsepower. A person who learns readily from cultural transmission, coordinates well with others, and participates productively in group efforts might outcompete someone with superior individual problem-solving ability but poor social skills. This shift in selection pressure enabled self-domestication. Once cultural knowledge became rich enough that individuals could thrive by learning from others rather than innovating independently, selection could favor pro-social traits even if they came packaged with reduced brain size. In resource-limited environments, these changes could accumulate into meaningful fitness differences across generations. The self-domestication hypothesis therefore links brain reduction to the same social and cultural changes explored in previous chapters, but through a different mechanism. Rather than brain reduction occurring because collective intelligence made large individual brains unnecessary, reduction occurred because selection for pro-social behavior in large groups produced smaller brains as a byproduct. The distinction matters. The collective intelligence hypothesis suggests brain reduction represents cognitive optimization, shedding unnecessary capacity once culture took over cognitive work. The self-domestication hypothesis suggests reduction represents social optimization, with brain size changes occurring incidentally as selection favored temperament over cognition. These hypotheses need not be mutually exclusive. Both processes could operate simultaneously, reinforcing each other. Cumulative cultural evolution reduced the cognitive demands on individuals, which relaxed selection pressure, maintaining large brains. Simultaneously, increasing group size and social complexity intensified selection for prosocial behavior, which actively reduced brain size through the developmental mechanisms of domestication syndrome. The result was brain reduction driven by both relaxed cognitive selection and active social selection, proceeding faster than either force alone would produce. Regional variation in brain reduction rates might also reflect variation in social organization. Populations that aggregated into larger, denser settlements earlier might have experienced stronger selection for prosocial behavior and thus faster self-domestication. The limited fossil evidence makes testing this difficult, but some studies suggest populations that developed complex agricultural societies relatively early experienced particularly rapid brain reduction. Critics raise several challenges to the self-domestication hypothesis. First, the domestication syndrome traits in humans are not as pronounced as in truly domesticated animals. Dogs show dramatic morphological changes from wolves. Domesticated foxes from the Siberian experiments developed spotted coats, curly tails, and radical behavioral shifts within just 40 generations. Human changes are more subtle. While we do show reduced facial robustness, smaller teeth, and decreased dimorphism, we lack the floppy ears, spotted coats, and extreme neoteny seen in many domesticated species. If we truly underwent self-domestication comparable to animal domestication, the physical changes should be more dramatic. Second, the hypothesis struggles to explain the specific timing. If social selection for prosocial behavior drove self-domestication, why did it not begin far earlier? Homo sapiens lived in social groups for hundreds of thousands of years before recent millennia. Even earlier hominins lived in bands requiring cooperation. If these social conditions suffice to trigger self-domestication, brain reduction should have begun much earlier, not specifically when the fossil record documents it.
Third, distinguishing self-domestication effects from other causes of brain reduction proves challenging. The metabolic efficiency hypothesis, discussed in earlier chapters, could produce similar patterns through entirely different mechanisms. Collective intelligence, reducing cognitive demands, could allow brain reduction without any self-domestication occurring. Multiple causes might produce similar outcomes, making it difficult to determine which factor actually drove the observed changes from fossil evidence alone. Fourth, the hypothesis requires that social selection consistently favored pro-social individuals across all populations globally. Yet human societies varied enormously in structure, size, and organization. Some remained small hunter-gatherer bands, while others formed agricultural villages and eventually empires. Some emphasized egalitarian social structures, while others developed rigid hierarchies. Did all these diverse societies exert selection for pro-social behavior in ways that produced convergent brain reduction? The global pattern of reduction suggests a universal cause, but the diversity of human social organization complicates simple stories about uniform selection pressures.
Despite these challenges, the self-domestication hypothesis remains compelling because it connects human brain reduction to the one feature that most clearly distinguishes Homo sapiens from other hominins, our hypersocial nature. We cooperate with strangers. We live in groups orders of magnitude larger than other primates. We tolerate crowding that would generate violent conflict in chimpanzees or bonobos. We have subdued our aggressive impulses sufficiently to function in cities of millions. These distinctive human social capacities emerged relatively recently in our evolutionary history, during the same period when brain reduction occurred. The hypothesis proposes a direct causal link rather than treating these as independent changes. The comparison with Neanderthals proves particularly instructive. These extinct hominins possessed brains equal to or larger than modern humans, yet they apparently lived in smaller, more isolated groups. If self-domestication fundamentally altered human temperament, making us more cooperative and less aggressive than our ancestors, this could explain both our evolutionary success and our brain reduction. We sacrificed some individual cognitive capacity, paying the price of domestication syndrome, but gained social abilities that proved more valuable for survival in the crowded, interconnected world humans created. The largest brains in human history belong to people who still operated primarily as individual cognitive agents. We have become smaller-brained not because we became stupider, but because we became friendlier, trading individual prowess for collective harmony. Whether this represents progress, adaptation, or loss remains open to interpretation. But if the self-domestication hypothesis proves correct, the trajectory seems clear. Humanity domesticated itself, and like all domesticated animals, we paid in brain size for the temperament we gained.
Chapter 4. From Neurons to Networks. 5,000 years ago, in the ancient city of Uruk, scribes pressed wedge-shaped marks into soft clay tablets, creating the world's first writing system. These cuneiform records tracked grain stores, livestock counts, land transactions, and tax obligations. For the first time in human history, critical information existed outside human memory. A merchant could die, but the record of debts owed to him would persist. A priest could forget the exact ritual calendar, but clay tablets would preserve it. The cognitive work of remembering shifted from neurons to artifacts. Consider what a person needed to know in deep prehistory, before the agricultural revolution and certainly before cities and writing. A hunter-gatherer required intimate knowledge of their local environment spanning hundreds of square kilometers. They had to recognize dozens or hundreds of plant species, knowing which were edible, which were toxic, which had medicinal properties, and when each would be available throughout the seasonal cycle. They needed to understand animal behavior, tracking patterns, migration routes, and hunting tactics for multiple prey species. They had to navigate without maps, often across featureless terrain, using landmarks, star positions, wind patterns, and subtle environmental cues. They needed to know how to construct shelters, make fire in various conditions, manufacture tools from stone and organic materials, treat injuries and illnesses, predict weather, and coordinate complex social interactions within their band. All of this knowledge resided in individual brains. While these foragers certainly shared information and learned from each other, they could not consult written references, ask specialists for help at a distance, or rely on institutional knowledge stored in organizations. Each person carried a substantial portion of their society's total knowledge base. Losing an elder meant losing irreplaceable information about rare plants, distant water sources, or techniques for handling unusual situations. The death of a skilled toolmaker might leave a gap in the community's capabilities that took years to fill as others learned to replicate lost expertise. Compare this to a person living in Uruk 5,000 years ago. This city dweller might know only a single specialized skill. A potter would know clay preparation, wheel throwing, kiln management, and glazing techniques, but might know nothing about farming, navigation, or hunting. A scribe would master the complex cuneiform writing system with its hundreds of signs, but might have no idea how to make the clay tablets he inscribed. A farmer would understand irrigation, crop rotation, and harvest timing, but might never learn to read, navigate beyond familiar fields, or craft anything more complex than basic tools. This extreme specialization was possible because knowledge became distributed across the population rather than replicated in each mind. The potter did not need to know how to grow wheat because farmers specialized in that. The scribe did not need to know pottery because potters supplied vessels. Each person mastered a narrow domain while depending on a network of other specialists for everything else. The total knowledge contained in Uruk's population vastly exceeded what any earlier band possessed. But each individual Uruk resident knew far less than a forager. Writing systems transformed the equation further. Once information could be stored externally, it became cumulative across generations in a new way. Oral traditions could preserve some knowledge for centuries, but writing allowed perfect replication. A mathematical insight discovered in Babylon could be copied and transmitted to Egypt without degradation. Astronomical observations accumulated over generations, enabling predictions impossible from a single human lifetime of observations. Legal codes could grow increasingly complex as each generation added refinements to inherited frameworks. This transition parallels what researchers observe in ant colonies, the most sophisticated examples of collective intelligence outside humans. Ants achieve remarkable cognitive feats through the interaction of many simple individuals, each following basic rules without understanding the system as a whole. Consider the leafcutter ants of genus Atta, which maintain elaborate underground fungus gardens. No individual ant understands fungus cultivation. Workers specialize in cutting leaves. Others transport leaf fragments. Others chew leaves into substrate. Others tend the fungus. Others defend against parasites. Each performs a simple task, but their coordinated efforts sustain a complex agricultural system. The brain size of individual leafcutter ant workers is remarkably small relative to solitary insects of similar body size. This reduction reflects their reliance on collective intelligence. A solitary insect must solve all problems individually, requiring neural circuitry for navigation, food recognition, predator avoidance, nest construction, and more. An ant worker in a colony can afford a smaller brain because the colony handles these challenges collectively. The worker needs only the neural circuitry for its specialized role, plus enough social communication ability to coordinate with nestmates. Research shows that when worker ants become reproductive queens, their brains can shrink by 19%, only to regrow if they revert to worker status, demonstrating that brain size tracks functional demands. Studies of different ant species reveal that more socially complex species with greater division of labor tend to have smaller individual brain sizes relative to body mass. Ocophilo weaver ants, which construct elaborate leaf nests through coordinated group effort, show this pattern. Formica garden ants, which tend aphid colonies like livestock, display reduced brain-to-body ratios compared to solitary relatives. The leafcutter ants, Atta, demonstrate the same trend. Their individual cognitive simplicity enables collective sophistication. The colony as a whole makes complex decisions about nest location, resource allocation, and defense strategy. But no individual ant possesses the neural capacity to understand these colony-level choices. Human societies evolved convergent patterns despite utterly different evolutionary paths and mechanisms. As human social complexity grew during recent millennia, individual cognitive demands could decrease because the group handled more tasks collectively. A city resident in ancient Mesopotamia could rely on the collective intelligence of thousands of specialists. When faced with a problem outside their expertise, they could consult someone who knew. The cognitive burden spread across the network rather than concentrating in each mind. This distribution of knowledge and cognitive labor creates efficiency at both individual and group levels. If external knowledge storage and social networks can compensate for reduced individual processing capacity, then smaller brains become viable without compromising survival or reproduction. Consider how memory itself transformed during this period. A pre-literate forager needed episodic memory for countless specific events.
where a particular fruiting tree stood, when certain animals last appeared at a water source, which roots grew near specific landmarks. Semantic memory held accumulated facts, which mushrooms were edible, how to track different animals, what stars indicated which directions. Working memory juggled complex navigation calculations, social relationship tracking, and multi-step problem solving, all without external aids. After the development of writing and complex societies, much of this memory burden shifted externally. Farmers consulted lunar calendars rather than memorizing seasonal cycles. Travelers followed maps rather than maintaining elaborate mental landscapes. Merchants recorded transactions in ledgers rather than remembering every exchange. The neural circuitry supporting these internal memory systems could atrophy because external systems had rendered it redundant. Yet human cognitive performance did not decline. By every measure we can assess, late Holocene and modern humans equal or exceed the cognitive capabilities of their larger-brained ancestors. We accomplish this through leveraging the collective knowledge of our entire species. A modern engineer designing a bridge uses mathematics developed over thousands of years by hundreds of contributors. She consults building codes refined through generations of trial and error. She relies on material science discovered by researchers who spent lifetimes studying stress and failure. No single person invented the bridge. Humanity did, collectively, across time. This accumulation accelerated as external storage systems improved. Early writing could preserve only simple information, grain counts, tax records, royal genealogies. Later developments allowed more sophisticated content, legal codes, mathematical proofs, astronomical tables, philosophical arguments. Printing made rapid replication possible. Digital storage now enables effectively unlimited information preservation and instant global transmission. At each step, the amount of knowledge accessible to individuals increased, while the amount each brain needed to retain decreased. Yet this trajectory raises questions about dependence and resilience. If modern human intelligence depends critically on external systems and social networks, what happens when those systems fail? A late Pleistocene human with their larger brain, deposited alone in wilderness, possessed the knowledge and cognitive capacity to survive. A modern human in the same situation would likely perish despite their PhD or specialized expertise. We have become dependent on collective intelligence in ways that reduce individual capability even as they enhance group achievement. Written records can burn, digital storage can corrupt, social networks can fragment. If the external systems that now carry much of human knowledge were disrupted, could our smaller brains compensate? Perhaps this represents not loss but transformation. The largest human achievement may be the creation of systems that allow intelligence to exist beyond individual minds and lifetimes. Our species-level knowledge accumulates and compounds across generations in ways impossible for organisms limited to individual cognition. Mathematical physics, molecular biology, and space exploration require cognitive resources that exceed what any brain could contain. Only through distributed intelligence across specialists and generations did we develop these domains. The brain shrinkage of recent millennia might mark the point where human evolution fully committed to this strategy. The largest brains in human history belong to people who still operated primarily as individual cognitive agents, carrying most necessary knowledge internally. As cultural evolution created alternatives, writing, specialization, dense social networks, and now digital systems, biological evolution eliminated the expensive neural infrastructure these alternatives rendered unnecessary. We became smaller-brained not because we became less intelligent, but because intelligence moved from individual to collective, from biological to cultural, from neurons to networks. This interpretation, however, competes with alternative explanations explored in other chapters. Chapter 3 examined whether self-domestication, selection for pro-social behavior in large groups, produced smaller brains as a developmental byproduct rather than as a response to reduced cognitive demands.
Climate hypotheses suggest that warming temperatures after the last ice age favored smaller brains for thermoregulatory reasons. These competing explanations need not be mutually exclusive. Multiple pressures may have operated simultaneously, each contributing to the same outcome through different mechanisms. What remains clear is the unprecedented nature of this transformation. No other species has externalized knowledge on the scale humans achieved during recent millennia. No other species reduced brain size during the same period when they radically expanded the cognitive resources available to their populations. Whether this trajectory continues, whether human brains will shrink further as our external knowledge systems grow more sophisticated, depends on whether we have reached some floor below which further reduction would compromise even our ability to interface with the collective intelligence we have created.
Chapter 5. The only primate that ages this way. Human brains shrink dramatically with age. Chimpanzee brains do not. This stark difference, discovered through an unusual collaboration between neuroscientists, anthropologists, and primatologists, fundamentally distinguishes our species from our closest living relatives. The research team assembled magnetic resonance images from 87 humans ranging from age 22 to 88, paired with scans from 99 chimpanzees spanning ages 10 to 51. The chimpanzee age range covered the full adult lifespan observed in wild populations, where few individuals survive past their mid-40s, though in captivity a few have lived into their 60s. The human range extended well beyond, into the decades where our species uniquely persists long after reproductive capacity ends. When researchers compared these images, measuring volumes of brain structures across both species and all ages, they discovered something unprecedented among primates. The human brains showed exactly what decades of research had documented: progressive shrinkage with age. Gray matter decreased, white matter declined. Ventricular spaces expanded as brain tissue pulled away from the skull. The cerebral cortex, involved in higher-order thinking, experienced more shrinkage than the cerebellum, which handles motor control. By age 80, the average human brain had lost 15% of the weight it possessed at its peak in early adulthood. The hippocampus, essential for forming new memories, showed measurable reduction. These changes accelerated after age 60, and no brain structure proved immune to the steady erosion of time. The chimpanzee brains told a different story. Across the full adult lifespan, from young adulthood to the oldest individuals in the study, chimp brains showed no significant volume loss. A 50-year-old chimpanzee possessed essentially the same brain volume as a 10-year-old adult chimp. The gray matter did not shrink. The ventricles did not expand. Even the most elderly chimpanzees, rare individuals who had survived well beyond typical wild lifespans in the protected environment of captivity, showed no evidence of the progressive atrophy that characterizes human aging. This finding was not subtle. The researchers measured it across multiple brain regions using standardized protocols. They accounted for body size, sex, and individual variation. The conclusion remained robust. Human brains shrink with age in ways that chimpanzee brains do not. Chet Sherwood, the anthropologist who led this research at George Washington University, called the findings surprising. As he noted during interviews while his 7-year-old daughter was being looked after by his mother, itself an example of the grandmother pattern he was studying, the relatively short evolutionary separation between our species should not have produced such complete divergence in brain aging patterns. This distinction extends beyond chimpanzees to all other great apes. Gorillas, orangutans, and bonobos all maintain stable brain volumes across their adult lifespans. None show the progressive atrophy characteristic of human aging. We are the only primate species whose brains shrink substantially as we age, the only ones whose neurons progressively die off in ways that compromise cognitive function, the only ones who develop age-related dementia with any regularity. Sherwood proposed that the answer lies in our unusual lifespan. Humans live far longer than other great apes, and more importantly, we live decades beyond our reproductive years. Female chimpanzees remain fertile until near the end of their lives. Human women cease reproduction around age 50, but routinely live into their 70s, 80s, or beyond. This extended post-reproductive lifespan appears unique among primates. Evolutionary biologists explain it through the grandmother hypothesis. Older humans who have ceased reproducing themselves can increase their genetic fitness by helping their adult children raise grandchildren. The benefit is quantifiable in traditional societies, where the presence of grandmothers significantly improves grandchild survival and nutrition. But this extended lifespan creates a biological challenge that other primates never evolved to solve. The human brain must continue functioning for decades beyond the lifespan that characterized our primate ancestors, potentially 80 years or more. Most neurons in the adult brain do not regenerate. When a neuron dies, it cannot be replaced. The brain must maintain a pool of neurons established during development for the entire duration of life. This represents a maintenance problem of staggering proportions.
The cellular biology of neurons explains why this challenge is so severe. Unlike most cells in the body, which divide and replace themselves regularly, neurons in most brain regions are postmitotic. They ceased dividing during development and persist as the same individual cells for decades. A neuron formed when you were 6 years old remains the same cell when you are 60 years old. It has accumulated 54 years of molecular damage, metabolic stress, and accumulated errors in protein production. Around age 40, even in normal aging, the myelin sheath that insulates nerve fibers begins to deteriorate, slowing neural transmission. The metabolic demands of maintaining neural function produce reactive oxygen species that damage cellular components. Proteins misfold and aggregate into toxic clumps. DNA accumulates mutations. Mitochondria decline in efficiency. Across decades, these insults compound. Other primates avoid the worst of this because they do not live long enough for the damage to become catastrophic. By the time a chimp's neurons have accumulated 40 years of damage, that chimp is near the end of its natural lifespan anyway. Natural selection never optimized chimp brains for lifespan extension because it never occurred in the wild.
Humans, by contrast, routinely experience the consequences of extended neural aging. The dementia that afflicts roughly a third of humans who survive into their 80s has never been reliably documented in any other primate species. Alzheimer's disease, with its characteristic accumulation of amyloid plaques and tau tangles, appears to be uniquely human, or at least uniquely common in humans. As neuroscientist Tom Pruess noted, while chimpanzees are different from humans, this does not make other animals useless as models for studying age-related diseases. Instead, the differences could help explain why humans suffer more from these diseases than other animals seem to. Against this backdrop of uniquely human brain aging across the normal lifespan, the additional shrinkage documented over the past 10,000 years takes on particular significance. We are discussing a reduction in maximum brain size across the population, affecting young adults as well as elderly individuals. A young adult human today possesses a smaller brain than a young adult from the late Pleistocene, before significant age-related shrinkage has occurred in either individual. This population-level reduction over recent millennia has no parallel in any other primate lineage that we can observe. Chimpanzees today possess the same brain size as fossil chimps from hundreds of thousands of years ago. Gorillas show no evidence of systematic brain reduction over time. Orangutans remain stable. Only humans display this pattern of recent rapid decrease overlaid on top of our unique pattern of age-related decline.
The combination creates a double burden unique to our species. First, we experience progressive brain shrinkage across our individual lifespans in ways other primates do not, a consequence of pushing neural maintenance systems beyond their evolutionary design limits. Second, we have experienced a population-level reduction in brain size over recent millennia that reduce the starting point from which individual age-related decline proceeds.
A modern human starts adult life with substantially less brain volume than a late Pleistocene human, then loses another 15% through normal aging. What drove this population-level reduction specifically during the Holocene while other primates remained stable? Unlike humans, other apes did not undergo the massive population expansion that characterized human history over the past 10,000 years. They did not develop agriculture, settle in permanent communities, or create complex states. They did not invent writing or external knowledge storage. They remained in essentially the same cognitive and social ecological niche their ancestors had occupied for millions of years. This stability provides a kind of control condition. If environmental factors like climate change drove brain reduction universally, we might expect other primates in the same environments to show similar trends. They did not. Yet we remain the only primate that experiences age-related dementia with any regularity, the only one whose neurons deteriorate badly enough with age to compromise function across entire domains of cognition.
We took the basic primate brain design and asked it to do things no primate brain had done before. Run for 80 years, support population sizes in the millions, interface with external knowledge systems. The modifications we made through biological evolution over millions of years solved some challenges but created others. Does recent brain reduction exacerbate our vulnerability to age-related neurodegeneration or mitigate it by reducing the metabolic burden of brain maintenance? The chimpanzee scans provide no answers, only confirmation that humans have traveled an unprecedented path among primates. Aging in ways no other species does, shrinking in ways no other lineage has, vulnerable to cognitive decline unknown in our closest relatives. The story of human brain shrinkage forces us to reconsider what progress means. For 2 million years, bigger brains defined our evolutionary trajectory. Then, precisely when we became most successful as a species, that trend reversed. Whether driven by warming climates, self-domestication favoring cooperation over aggression, or our shift toward collective intelligence stored in culture rather than neurons, we lost 10 to 15% of our brain volume in just a few thousand years. Yet, we split atoms and mapped genomes with these smaller brains. We became the only primate whose brains shrink both across evolutionary time and individual lifespans. The only species vulnerable to age-related dementia. The only ones who externalized knowledge so completely that intelligence became distributed across networks rather than concentrated in skulls. Perhaps the measure of a species is not the size of its brain, but what it does with the intelligence it creates, individually and collectively. Thank you for listening.